there is a diversity among different animal groups or different animals. Over 1 million species of animals are described so far. So there is a lot of diversity among all different groups of animals. So how do we remember, how do we study the characters of these animals? So for this, a classification is very necessary. So in spite of differences among different organisms or different animals, there are some fundamental features which are common among different individuals, such as arrangement of body cells. We call it as levels of organization, symmetry, coelom, organ systems like digestive system, respiratory system. Based on these fundamental features or characters, we have classified the animals into different groups. So let us find out what are these fundamental features with which the animals are classified. So we go with the first, levels of organization. So in organizational hierarchy, every animal starts their life with a single cell. Cell becomes many cells called tissue. Tissue become an organ. Organ become organ system, and so organism. So the first level of organization is cellular level. In these animals, they have many cells in the body, and the division of labor is within the cells. Cells only perform all different functions, but there are no tissues. Because there is no coordinating cells like nerve cells on the body, as a result, the organization will remain at the level of cells. So this is called cell aggregate type of body form. So this kind of cellular level of organization is found in phylum Porifera. So these are sponges where the organization remains at the level of cells. There is division of labor among the cells. Next, in phylum Cnidarians and in Ctenophora, cells are combined to form tissues which are specialized to perform different functions. So this is the second highest level of organization in the animal, with the tissue level of organization, which is found in Cnidarians and Ctenophores. Next, in phylum Platyhelminthes, this is the first triploblastic phylum where tissues are combined to form organs which are performing a set of functions. This is called organ level of organization. And from Aschelminthes onwards till chordates, this is from phylum Aschelminthes to chordates, they have organ system level. So different organs combined to form organ systems, and these organ systems are specified for performing some physiological functions. This is the highest level of organization. Even in organ system level organized animals, there is a lot of differences or diversity among the various organ systems, or what physiological functions that organ system will perform. So let's take up an example of a digestive system and circulatory system, how it, what kind of physiological functions it performs and how it developed in different organisms or different animals. Digestive system. In Platyhelminthes, they have only one opening to the alimentary canal. This is gut. Gut is alimentary canal. There is only one opening which acts for ingestion as well as for egestion. This is called two-way gut, and this is called incomplete gut. Next, this is what you find in Platyhelminthes. Next, in Aschelmanthus took chordates. From Aschelmanthus to chordates, in all this, gut is with anterior opening called mouth for ingestion and opposite is anus for egestion. There are two openings. This is called complete gut. So complete gut is also known as one-way gut. This is called two-way gut. So this is from Aschelmanthus to all chordates, they have complete gut. This is how the digestive is different in different organ system level organized animals. Next, circulatory system. In some animals, the circulatory system is called closed, because this is where the blood flow in closed vessels, blood vessels like arteries, veins and capillaries. The blood keeps moving in all these closed vessels. It's called as closed circulation. And closed circulation is in phylum Annelida. Annelids have closed circulation among the non-chordates, and closed circulation is found in cephalochordates. Cephalochordata is with closed circulation, and remaining all vertebrates. Vertebrates have closed circulation. This is how you can see closed circulation, where the blood flow in blood vessels, closed vessels. Next, there is open circulation, open type. In open type of blood circulation, blood flow in body cavities, right, spaces, right. So this is called open type. Open type of blood circulation is found in phylum Arthropoda, phylum Mollusca, phylum Echinodermata and phylum Hemichordata and Urochordata. Urochordata is subphylum. In all this, circulation is open type of circulation. Next, molluscans. Just remember, all molluscans have open circulation except cephalopods. Cephalopods are the molluscans which have closed circulation, which includes your cuttlefishes, squids, octopus. All are placed under cephalopoda. Except cephalopoda, in remaining all circulatory system is open, but they are closed in those three. So these are the levels of organization. Next, second important or the fundamental character is symmetry. So first, just look at the definition of symmetry. Symmetry is nothing but arrangement of body parts on opposite sides of a dividing plane or on sides around a axis. I'll just explain you in a simple terminology what is meaning of symmetry. Symmetry meaning is those all the animals which have a proper body shape, they have a central axis in the body. So all the body organs are arranged on either side of the central axis or around the central axis. If the animal is cut along that central axis, animal can be divided into two equal halves, and those two equal halves are called mirror images. We call them as antimirrors. If any animal can be cut into two such antimirrors, we call the animal is symmetrical. When we cannot divide the animal into two antimirrors, it's called asymmetrical. Fine. So like asymmetry is most of the sponges. Most of the sponges are asymmetrical. We cannot divide the sponge into two equal halves because they do not have a proper body shape. Next, we have two different types of symmetry to study here. One is radial symmetry and bilateral symmetry. Let us discuss what is radial symmetry. First, radial symmetry is exhibited by those animals which are either cylindrical in shape or umbrella shaped. So you might have understood already what I'm going to talk about. Cylindrical like hydra, right, is an example. There are some triploblastic animals do exhibit this, but in general cylindrical like hydra, umbrella shaped like jellyfishes. So these are diploblastic animals. They generally exhibit this radial symmetry. Next, some things that you have to remember here is, in radially symmetrical animals in general, I'm talking about without any exceptions, in general, in radially symmetrical animals, they do not have any anterior, posterior, dorsal, ventral, lateral surfaces. They have anterior opening, which is acting as a mouth for ingestion and anus for ejection, which is called oral end. This is called oral end. And the opposite end is called as an aboral end. That's it. They have oral and aboral ends. Now, this is the axis, dividing axis of the body, which is passing from oral to aboral. Right? Now, all the body organs are arranged around this oro-aboral axis. Now, if the animal is cut along this oro-aboral axis, we can divide it into two equal halves. We can cut like this, or you can cut the animal like this, or we can cut the animal like this. Any plane passing through this central axis can divide the animal into two antimeres. So the symmetry is called radial symmetry. So in radially symmetrical animals, please do remember this point. In radially symmetrical animals, any plane passing through the central axis of the body can divide it into two equal halves or antimeres. That is radial symmetry. I mean, we can divide the animal into two identical halves along many planes, but the plane should pass through the main axis or central axis of the body, which we call it as principal axis. Right? So this is exhibited by this particular symmetry is shown by cnidarians and ctenophores. There is one exception, echinoderms also exhibit radial symmetry. In the last, we'll talk about echinoderms, why they exhibit this symmetry. Next, bilateral symmetry. In all these bilateral symmetrical animals all are triploblastic. All triploblastic animals will exhibit bilateral symmetry with one or two exceptions. But in general, all triploblastic animals exhibit bilateral symmetry. Next, among the animals with symmetry, which is the most advanced symmetry? Animals of bilaterally symmetrical animals are more advanced compared to animals of any other symmetry. Reason is, all these bilaterally symmetrical animals are well cephalized. They are cephalized, meaning is there is a process called cephalization. Development of sense organs and nervous system is present at the anterior end of the body. This is called cephalization. Because of this cephalization, bilaterally symmetrical animals are more advanced than the animals of any other symmetry. Got it? Now, in bilaterally symmetrical animals, any triploblastic animals, we can take an example. In the NCERT book, crab is given as an example, just I'm taking it crab. So, in bilaterally symmetrical animals, this is the median axis or median sagittal plane or central axis of the body, or we can say that the principal axis of the body. Now, if the animal is cut along the principal axis, the animal can be divided into right and left halves. So in my diagram, your right will become left, left become right. So for that reason, I wrote in that way. So if an animal is cut along its central axis, that is principal axis, we can divide this into two right and left halves. We call those two are two lateral halves or two laterals. So the symmetry is bilateral symmetry. So in bilaterally symmetrical animals, only along one plane we can divide the animal into two equal antimeres. And whereas in radially symmetrical animals, any plane passing through the central axis can divide. That's the difference. Remember, if a definition comes, sometimes we get confused. What is the symmetry? whether it's a radial or bilateral. Remember, in radial symmetry, many planes or any plane passing through the central axis of the body will divide. But in bilateral symmetry, only one plane, that is median sagittal plane, passing through the principal axis can divide the animal into two right and left halves. We call them as antimeres, which is called bilateral symmetry. Now, no animal in the animal kingdom will exhibit or will show a perfect symmetry. This is one point. Next, the second thing what you have to remember is, while classifying the animals or while placing the animals in different groups, always we consider the symmetry exhibited by the animals in their embryonic stages but not in the adult. Like an example is your echinoderms. If the question is asked what is the symmetry in echinoderms the symmetry is radial symmetry, starfish. But echinoderms are not diploblastic, they are triploblastic. During larval stages, echinoderms are bilaterally symmetrical, and in the adult they exhibit radial symmetry. Echinoderms are placed under grade bilateria, but adult will show radial symmetry. Next, one more example like snails. Snails is a molluscan. Mollusks are with triploblastic and bilaterally symmetrical, but snails are asymmetrical in the adult. So this is we call primary symmetry and secondary symmetry. Primary symmetry of echinoderms is bilateral, secondarily they are radial. Next, snail also same thing. Primary symmetry is bilateral, secondarily it is asymmetrical, right? So this is symmetry. Another important character for the classification is the formation of germinal layers. When once many cells are formed, cells are specialized to form different tissues and organs. Before that, the cells will get arranged into different layers. So in some animals, there are only two layers are formed. This is called diploblastic body organization, where the outer layer, this layer is known as ectoderm, and this is inner layer which is known as endoderm. There are only two layers are formed. The cells from these two layers are they specialized to perform or to form different tissues which make different which perform different functions. So in tissue level organized animals, like mostly in Ndarians, you have only two layers, that is outer layer is ectoderm, inner layer is endoderm, and this is what the central cavity that is surrounded by this endoderm. Now the question is, what is present in between these two layers? In between these two layers is known as mesoglia. So mesoglia is non-cellular. It do not have any cells, but it is made or it is composed of gelatinous or gel-like substance, which is mostly it is filled with carbohydrates. So this is a diploblastic body organization, one of the important character for classification, and this is found in your phylum Cnidaria. Phylum Cnidaria and ctenophores are with two layers. Next, the another interesting thing is triploblastic, where between the outer ectoderm and inner endoderm, this is the third layer. The third layer called mesoderm is developed. So formation of mesoderm, right, is for the first time in Platyhelminthes. So Platyhelminthes is the first phylum which is a triploblastic phylum with mesoderm. Mesoderm is concerned with muscles and other structures. So mostly in triploblastic animals there are three layers, where outer ectoderm, inner endoderm, middle mesoderm. Cells become specialized structurally and functionally from these three different germ layers and form various organs and organ systems. So this triploblastic nature starts from phylum Platyhelminthes. So from Platyhelminthes to chordates, from Platyhelminthes to chordates, all are with triploblastic body nature, right? Next, another interesting feature for classifying the animals is coelom. So first understand the definition of coelom. What is it? So coelom is the body cavity which is present between the alimentary canal and the body wall. So I can say that coelom is the space which is between the body wall and gut, alimentary canal, that is known as coelom. So like you just compare, if any triploblastic animal you take, right, if this is the entire body wall of the animal, and imagine in the middle this is gut called alimentary canal, right? Now this space between the body wall and the alimentary canal is called coelom, right? Now you just compare this to understand. Compare this like a pen. You have pen, that is outer covering of the pen, and in the middle the refill. Now the space between the outer covering and refill is nothing but the coelom. Just you compare the animal in that way. So alimentary canal is surrounded by cavity. So that is the reason coelom can also be called as perivisceral cavity. It's present surrounding the visceral organ like alimentary canal. So now how exactly coelom is developed? So coelom is exclusively formed from mesoderm. There are animals where between the ectoderm, this ectoderm, it generally forms it lines the body wall, and this is the endoderm. This endoderm lines the alimentary canal or gut. This is alimentary canal, if you just take a section of the animal. Now in between these two layers is the mesoderm. Now here between the alimentary canal, that is gut, and body wall, there is no space. It's completely occupied by mesoderm. So there is no cavity. So this is what you call a coelom. So those animals where there is no body cavity, the space is completely filled with mesoderm that forms parenchyma tissue or mesenchymal tissue, is known as Coelomates, or this condition is called as coelom. This coelom condition can be seen in phylum Platyhelminthes. So in Platyhelminthes there is no body cavity. It's completely filled with mesoderm between the body wall and gut. So this is what he called coelom condition. Next, just look at into another, like in the phylum Aschelminthes. This is the phylum Aschelminthes. We called roundworms. In that, just look here. This is the outer layer, that is ectoderm, that forms the body wall, and this is the endoderm that lines the gut called alimentary canal. Now there is mesoderm. All this red is mesoderm. Mesoderm is present in the form of mesodermal pouches. These are mesodermal pouches. So you can see there is a space. This space is coelom, which is not a true coelom. It's called pseudocoelom. Now this is a space. This space is pseudocoelom, or this is called pseudocoel. This is a pseudocoelom. Cavity is present, but it is not a true cavity. It's called pseudocoelom or false body cavity. Now this pseudocoelom or false body cavity is also filled with a fluid which is called pseudocoelomic fluid. Now this pseudocoelomic fluid is advantageous to the animal in many ways. Like first, the pseudocoelom acts as hydrostatic skeleton that maintains rigidness that gives rigidness to the animal body. Number two, it provides space for the movement of internal organs, and also it provides space for lodging the young ones. Like let us take an example of Eukarya bancrofti, which is a filarial worm. it produces, it's called as ovoviviparous. It gives birth to the larvae. The larvae are present inside the body of the female. So for providing space for the larvae, pseudocoelom helps. So pseudocoelom is providing space for lodging the young ones. So these are the advantages in pseudocoelomate animals. Now, one more interesting character for pseudocoelomates is, in pseudocoelomates, generally circulatory system is absent. Roundworms do not have circulatory system. So, because of absence of the circulatory system, those functions of the circulatory system are compensated by pseudocoelom. That means I can say that pseudocoelom is also involved in transport. So these are the advantages of pseudocoelom. Fine. Now the question is, what is a true coelom? How we call the coelom is a true coelom, which is called eucoelom. Let us see what exactly pseudocoelom is, or true coelom is. Now same, this is the outermost layer, that is ectoderm, and this ectoderm line the complete body wall from outside. This is body wall. So body wall is completely lined by the ectoderm, and this is the gut we call alimentary canal, which is lined by endoderm. This is endoderm. Endoderm covers the gut. Now there is a third layer called mesoderm. Just look here, this red covering is mesoderm. This is mesoderm. This is mesoderm, that is towards the body wall, and this is the mesoderm. This is the mesoderm towards the alimentary canal, gut wall. So actually this mesoderm forms muscles on both body wall as well as on the gut. Now this space is called coelom. I am just calling it as eucoelom, that is true coelom. Why? Because this coelom is lined by mesoderm on both sides, and this mesoderm lining the coelom on both sides is called as peritoneum. This is peritoneum. Mesoderm lining the coelom on both sides is called peritoneum. Such a coelom is called true coelom. Now just come back to this pseudocoelom. Is there any mesoderm covering on both sides? No. So it's a pseudocoelom. It's a false cavity. But whereas this is the true body cavity or a true coelom. So true body cavity or a true coelom is a coelom which is lined by mesoderm on both sides. Right? So this is what you call it as eucoelom. Now eucoelomates are annelids. First eucoelomates are annelids. Annelida, arthropoda, mollusca, echinodermata, hemichordata, chordates. All these are true coelomates. We call them as eucoelomates. Next, for your reference, these eucoelomates, we have further classified them based on the method of formation of mesoderm. So that much information is not required. Just I will tell you the two types of eucoelom. Remember this. This eucoelom is further called as schizocoelom. Schizocoelom. So this is based on the formation of mesoderm, how exactly the mesoderm is formed. Based on that, the true coelom is further classified. This is the first one, is called schizocoelom. Schizocoelom type of coelom formation is found in annelids. So remember, in Annelida and in Arthropoda and Mollusca. In all these three, we have schizocoelom. Next, there is one more true coelom which is known as enterocoelom. I am just rubbing this part here. One more true coelom is known as enterocoelom. Enterocoelom type of coelom formation, which is also a true coelom, is found in your echinoderms. Hemichordates and chordata. In echinoderms, hemichordates and chordates, we have enterocoelom, and schizocilum is found in Annelida, arthropod and mollusca. Both schizo and enterocoelom are true coelom. What is a true coelom? Which is lined by mesoderm on both sides. Fine. True coelom is filled with coelomic fluid. Remember, coelom is filled with the fluid called as coelomic fluid. Same. The coelomic fluid acts as hydrostatic skeleton, and it provides space for the contractions and the movement of internal organs, and also this true coelom provides space for lodging the young ones in case of viviparous organisms or viviparous animals. Right? Now, one more interesting point or one more special character what you have to remember is the functional body cavity in arthropods and molluscans. The question would be asked, remember, the functional body cavity in arthropods and molluscans is known as hemocoel. Remember, the functional body cavity in arthropods and molluscans is called hemocoel because they have open circulatory system. Entire body cavity is filled with blood, so it's called hemocoel. Hemocoel is the functional body cavity in arthropods and molluscans. So clear? So this is coelom is also one of the specialized or important character for classification of animals. Next, like in animals, like annelids in phylum Annelida and phylum Arthropoda, body is divided into segments. This is what he called segmentation. Now, when in annelids, segmentation is both external as well as internal. In annelids, the segmentation is both external as well as internal segmentation. In annelids, they have both external as well as internal. Whereas in arthropods, segmentation is only external segmentation. Remember these things. Even segmentation, the animals, the phyla which are segmented are annelids and arthropods. Next, when you talk about like earthworm, in earthworm, which is an annelid, the segments, these are the body segments. All these are body segments. These are all body segments which are linearly arranged. And in these body segments, at least every segment represent some parts in a linear repetition. There is a linear repetition of at least some parts in all these segments. So for that reason, the segmentation is known as metamerism. So this is metamerism or metameric segmentation because they show linear arrangement of segments where there is a repetition of at least some parts in the segments you can see, which is called metamerism. Right? Next, the last character for the classification is we have notochord. Notochord is derived from mesoderm. It is mesodermal in origin. Notochord is placed on the dorsal side. It is a flexible elastic rod like structure. That flexible elastic rod like structure is called as notochord. Animals with notochord are called chordates. Animals without notochord are called as non-chordates. This is also one of the interesting feature for classification. Right? Notochord is present on the dorsal side. Right? It is derived from mesoderm. Animals with notochord are called chordates, without are called as non-chordates. Right? So these are the important characters for classifying any organism, or mostly for animal classification we use these are some of the basic characters for classification.